We're going to talk about prospective research design. Just give you a little background on myself. Prior to me coming here at UT Southwestern, I've been here for almost 2.5 years. I was the vice chair of research for plastic reconstructive surgery. I did a lot of research there. Coming here, I sort of switched gears. I'm doing about half-time administrative work and half-time clinical work here at UT Southwestern, Dallas, Texas. All right, so the first question to the audience is, why do we conduct research? One of the answers, A, is for fame and fortune. B, we have to fulfill residency fellowship requirements. C, to legitimize our profession. D, to disseminate information. What do you guys think it is?
You can put your answers in the chat.
E. What's that?
They can put their answers in the chat if they want.
All right, let me see if I can even pull up the chat. Where's the chat?
Q&A in that corner.
Okay. All of the above. Okay, so fame and fortune. So that was what Youssef Mustafa said. I'll remember that name. Just teasing. The reason really why we do it is to conduct or to disseminate information. That's really the only reason we should be publishing, because if you hold on to information and knowledge and it's not disseminated, it's worthless. And so you have to share information, and we do that in a kind of a more rigid fashion, and that's research, and that's part of research design. But ultimately, I know all of you won't be researchers, but you have to be educated consumers, because you're going to be reading the literature for the rest of your professional life. And so you have to be educated on how research is conducted in order to understand it once it's published. So I totally get it. I would say 95% of you will never publish a study or will conduct any research of any kind. But all of you will be reading the research, and to understand what you're reading, the best way is to conduct it. So if you have an opportunity to do so, I would suggest that you do that. So let's start with some working definitions. So what does randomization mean? And essentially, there's two kinds of randomization. Sampling, meaning in a population, you randomly select people in that population. Second way to think about randomization is allocation, is allocating a group of patients to one group versus the other. That's randomization in a nutshell. By the way, I'm going to talk kind of high level at some of this stuff. If you want more detailed information, I do suggest you read more about how to conduct research. There's a lot of resources available through the ACFE's website. It really does a really nice job. And Liz Ansert's been really very pivotal on the research committee this year, who's been really trying to put all that together. The next thing you want to look at as far as definitions are variables. So the independent variable, that's what your intervention actually is. It's independent. It's your treatment. It's your experimental activity, whatever you're manipulating. And the dependent variable is the response that you're looking for based on the independent variable. So based on the treatment or the intervention, that is your dependent variable. Further, when we look at definitions, there's two essential groups if you're doing comparative studies. There's the experimental group, which gets the intervention, and there's the control group, which doesn't get the intervention. Doesn't mean they don't get anything. Typically, it's the standard of care or some other treatment that's not the treatment that you're trying to study. The next definition is validity, and the validity really just says how reliable are the results that you're getting, and are you controlling for things like bias? Are you controlling for confounders that may influence the results? All of those things are considered validity. And then blinding. So blinding is important. Sometimes you can't blind for certain trials that you're working on, but a single-blinded study is when it's the subject, meaning the patient, in our case mostly, or the examiner is blinded. One or the other is blinded. When it's double-blinded, both the subject and the examiner are blinded, meaning they don't know what the intervention, if they're actually applying the intervention or it's a control drug, for example. Also, when you look at studies and when you plan out studies, they can be single center, which is the vast majority of studies, or it could be multi-center. And multi-center, all that means is that more than one site's involved in the study, and they're all sharing the same protocol. And we're going to talk about protocol in just a few minutes. And then there's the direction study, which is the point of this lecture tonight. Is it prospectively collected, meaning are you going forward in time, or is it retrospectively collected? So a lot of the papers that Matt presented on Taylor fractures were retrospective. In fact, I think all of them are. And so retrospective means that whatever has interventions been done, you look back in time to see what those results are. And from the more rigid design perspective, prospective studies are preferred over retrospective studies because you're not controlling for the biases or the confounders that you can in a prospective study. You guys have all seen the EBM pyramid, and by the way, there's lots of versions of this. So be careful on how you look at which version that you're looking at when you look at evidence-based medicine pyramid. The top may not be the same as what you're used to seeing, or the bottom may not be what you're used to saying. But typically what you see is this, and this is the kind of the widely accepted EBM pyramid for clinicians, not for basic scientists, which often is the other pyramid. Or, epidemiologists use the other pyramid as well. So at the very bottom of this pyramid is animal bench studies, then you go to expert opinion, and then case series and case reports, and then retrospective comparative studies. In the foot and ankle literature, predominantly, I would say probably 80+% of what you're reading are retrospective and sometimes non-comparative studies. As you continue to climb the EBM ladder or the pyramid, the single center randomized controlled trials. So those are single sites, randomized, as we discussed. It's usually to allocation, not by selection of subject, a random selection of subjects within a population. And it's controlled, meaning it's got some other therapy. You have your experimental therapy, and then you have your control group. And then as you continue to climb, there's multi-center randomized controlled trials. And that's just involving more than one site. So it could be two sites, and that's still considered multi-center. At the very top are meta-analysis and systematic reviews. You have to be careful when you look at the literature. When somebody says it's systematic review, it's just a review of the published literature in that space. The true meta-analysis actually does an analysis of all, hopefully, prospective studies, not all the time. Sometimes retrospective studies are mixed in there as well, but you actually do a statistical analysis of the data that's presented. And that's a true meta-analysis. And that's really the peak, because it synthesizes all of them. Now, all of these levels have their drawbacks, and some are more detrimental to understanding what the outcome, if it's a true outcome or not. But as you continue to climb the ladder, some people would argue that the meta-analysis is actually a poor way of looking at evidence, because meta-analysis typically involves heterogeneous populations, various different designs of how it was conducted, the research was conducted. So some people say that's not very good. Others will argue, you guys hear this, I hear this at meetings all the time. This was an RCT, meaning that it was some sort of gold standard. It's not. RCTs are fraught with problems. And we'll talk about that in just a minute as well. Okay, so when we look at research design in general, there's the quantitative, which is the kind that I think are the strongest, which is you test the effect of a drug, biologic, or drug versus a control, standard of care, or placebo. And then there's more qualitative, and we really don't do a lot of qualitative research in surgery in general. That's just simply not done. And by the way, I just want to make a comment that I made. I just want to clarify so the students understand this, is that it's not just in foot and ankle surgery that there is a paucity of more prospective studies. It's in all aspects of surgery, every aspect. In fact, I'm in the Department of Plastic Surgery, and they're notoriously bad about conducting high-level research or evidence. Most of them are surgical retrospective outcome studies and also technique papers. So it's not just us, it's pretty much everyone. So we're going to focus really on the quantitative studies. And so this is just a simple algorithm. So the first question you ask is there a treatment? And I'm going to say yes. You go down the right-hand column. Is the treatment tightly controlled by the researcher? If it's no, then it's called quasi-experimental, which I see a lot of that's published that supposedly are true experimental, but it's not. If it's yes, will a randomly assigned comparison control group be used? And if the answer is yes, is the original sample randomly selected? That's the ideal, but that's not necessarily how things work. Because patients sometimes consent to participate, sometimes they don't. You can't force them to, so it's not truly random selection. And then if that's true, then it's experimental study. If you go down the other algorithm, is there treatment? And the answer is no. Is the primary purpose examination relationship? If the answer is yes. Will the sample be studied as a single group? If the answer is yes, then it's a correlational design, meaning you look, and we'll define those in just a minute. So correlational design studies examines linear relationships between two or more variables, determining the type and the strength of that relationship. And an example I always give, you guys may have heard this story before, but the classic correlational piece of research is the fact the story about the storks in Florida. There's a correlation between stork sightings and live births in Florida. So we know, and your sort of mind jumps, you're like, we've all taught with those stories of the stork bringing in babies, but we know that's not true. But statistically, there is a correlation there. So you have to put things in context to truly understand what you're reading. These are just some examples of correlational studies, describe relationships, which is typically what we do in foot and ankle research. We rarely try to predict relationships, although that's been done. And those are usually very large populations that you're looking at when you're trying to do some predictions. And we don't do theoretical research in our field. So if we go back to the true experimental design, you look for a cause and effect. And these are relationships. It's not a causality, meaning one didn't cause the other. But what happens is you're looking for an outcome that's different than what you would normally expect without the experiment being provided. So that's what you're looking for in experimental design. In the randomization, as I discussed earlier, your patients are typically randomized into treatment or control groups, and control can be a placebo, no treatment, or usual standard treatment of care, standard of care. And this is typically what you see. You have a pre-test. So you measure, let's say, a bunion angle. You do a chevron osteotomy, and that's the treatment. And then you measure the angle again in the post-test. Or another example is pain before treatment. And then let's say that example that Matt presented, which was the talar fracture. So you do a visual analog scale, for example, for pain for the patient, and then you treat it, and then you see what happens after the treatment, and you could do it at multiple time points. And then you compare it to another group that didn't get any treatment. Now you can see in the example that Matt gave, it's probably not very ethical to reserve treatment for that kind of fracture without actually treating it. You're not going to randomize that patient, say, oh, we're not going to fix your talus. That's rarely done. Sometimes it is done. I think the classic study is for knee scopes. And what they did is one group was randomized for true knee arthroplasty, and the other group was randomized with 2 small incisions, and nothing was done. The interesting thing about that study, and that's a great study, and I'm not sure what the ethics of that is, but the outcomes were no different, which I think is fascinating. So the control, which didn't get the intervention, which was the arthroscopy, showed no better benefit than the patients that, or showed no worse benefit than the patients that actually got treatment. Interesting. There are other kinds of experimental studies. This is called a pre-experimental study, where there's only a post-test analysis with the comparison group. And this is what I'm talking about here. So the patient got some treatment, and there's a post-test. So there's no pre-test done. There was no examination of their baseline prior to the treatment being administered. And then they just got a post-test done. But it is still compared to another group that did not get the treatment, but just got the post-test exam, whether that's a X-ray outcome, pain, quality of life. It could be lots of things. Here's another example where there's no control group whatsoever. So you do a pre-test, you do whatever your intervention is, and there's a post-test. And then you just compare the pre versus post in a single patient population group. And you see that in retrospective designs a lot of the times for example, pain in a patient. There's no control group, but they just tested before the patient had the intervention. They had the intervention, then they examined, for example, pain after their rehab period. And then there are other kinds where there are multiple groups. So there may be a different dose of a drug, would be another experimental group. So there's two doses being compared to a comparison, which is placebo for a drug. So there's lots of different permutations of this. And depending on what the question that you're asking, and by the way, doing more than more than two group, the experimental versus the comparison, and adding another group, that adds to the length of the study typically and the cost if it's a robustly designed studies. Then there's always sampling, meaning that you're taking multiple measurements over time. So you have multiple time points where you're taking, for example, a pain measurement and prior to treatment and then after treatment. What makes these multiple time series difficult is the statistics of it. The design is relatively simple, but the patient then is required to have multiple contacts with the research team, the coordinator, or the investigator. So it could become a little bit more cumbersome. So I just want to make some general comments here. No study design is perfect. I just want to make that clear. There is no gold standard. There's idealized, but it just simply doesn't exist, because what we're dealing with, especially the kind of research we do it on humans. There's so many unique variables within each individual, and the host is so complex, and the environment is very complex for that patient, where they live, what kind of access to healthcare they get, and the environment that you're conducting the research also plays a role in that. But what I want you guys to hear clearly is that doesn't mean you just dismiss all research. You have to develop your own tolerance to design flaws that are inherent in all studies. And you guys have your own level of tolerance. It's probably pretty high tolerance because you don't know too much about research until you get really, skilled at this. But then you have to figure out, does the design flaws make the research outcome fatal, meaning that it means nothing because there's so many flaws inherent in the research design, and you have to kind of figure that out. All studies contain biases, and the rigid rules and regulations and guidelines around research design help you try to limit these biases. So for example, in our profession, there's a lot of case series by a single surgeon, right? You see that a lot. Oh, we did 40 of these, did 50 of these, and they publish the results. And the results may be good, bad, or indifferent. But the point is, a single surgeon did that. So that's in that surgeon's hands. Those were the outcomes. Well, my hands are different than Liz's hands or Matt's hands. My experience is different than Liz's hands or Matt's hands. So for me to extrapolate those results to what I can achieve in my circumstances is going to be different. So that's the major, to me, the major problem with these retrospective large case series. Like, oh, look, I did 100 of these. I'm like, okay, that's great for you. Now you know how well you do with these. What does that tell me about how well I'm going to do with these? And so that's the biases that I'm going to talk that I talked about. And they can provide all study designs. When you do statistics, it's all about probabilities. It's not about absolutes. So it tells you that you're more likely to have that the truth is real, or the outcome is real. It doesn't tell you that it's absolutely real. It tells you within statistical probabilities, whatever you found is a real finding. There are no absolutes in research. There's only absolutes in mathematics. You guys know that. There's no absolutes in science of any kind. It's a little easier for basic scientists when they're doing animal studies, because, for example, if you do rat research, I did rat research for 2 years when I was an undergrad and post-undergrad. And these were Harlan Sprague-Dawley albino rats, genetically identical. They're housed identically. They were fed identical food. The same amount of water. So then you can remove all the individual biases for those kinds of animal studies. And that's why if you read animal studies, the number of subjects in each study, number of rats in this case in each study, are so small, yet they can find significance because those biases, those individual biases, are all washed out, because there's not very much, if any, biases inherent in the treatment groups that you're looking at. Like I said, there are inherent issues with all study designs, including prospective randomized controlled trials. The biggest issue for RCTs, in my opinion, is when you do an RCT, and I've done many, what happens with this is that you have to try to really hone down on your patient population, and you really have to try to get a homogeneous patient population. And you have to provide them exactly the same care, exactly the same treatment regimen, and all of that stuff. What that does is it creates a high, what's called internal validity, but really poor external validity. External validity is its applicability to someone or somewhere else. That's what the really the major flaw of prospective randomized controlled trials are. This is where multi-centers can help, because then you're saying not only in one location are these findings valid, but it's also valid in other locations, and it kind of washes out the differences that may be there. Whenever you look at anything, for example, outcomes of Taylor fractures, you can't just rely on one study. There are pivotal studies and there are seminal studies that suggest something novel and new and provides more information. But you have to look at the body of literature to come to a conclusion. It's never about one study. And so when you look at that body, some of it refutes somebody's findings, some of it supports somebody's findings. And what happens with ultimately when you make a decision or when there's consensus is that there's more papers that support a certain intervention versus another. And that's why that intervention is adopted. It's never 100%. So when you get started when looking at or designing prospective studies, I would just recommend you follow this guideline. So the difference between a proposal, which you've heard of, and a protocol, they're two different things. Proposals are arguments of relevance to achieve essentially funding. And that's like for NIH, DOD, ADA, or whatever kind of funding that you're looking at. And so you're writing a proposal, but a protocol provides how to step-by-step execute the actual study that you want. And it's often the template that you can use it as a template for IRB submission, because in your protocol, it should have all of those things that the IRB is looking for. So when you look at protocols, this is kind of how you go about doing it. You formulate a research question or state a purpose. You review the relevant literature. This is important. You need to look at what's been published before and what holes there are in the knowledge, where the knowledge gaps are. You develop a framework of how to execute this. You formulate objectives, questions, and hypotheses. You define the variables, identify the limitations of the study. You select research design. Whether it's prospective, retrospective, multi-center, or single site, whatever. You define the population and the sample size that you're looking for. You select the method of measurement. So whatever outcome you're looking for, you have to find the tool that can accurately measure that outcome. And this is where sometimes they fail, where there's two major failures in prospective studies. One, an operational definition that is not agreed upon by everyone. Meaning, let's say cutoff, what's considered high hemoglobin A1C? Some people would say 8, some people would say greater than 7. So if you can't find that operational definition, what is peripheral neuropathy? How you define that? Is a lack of Weinstein? Is it vibratory sensation? What is it? And then the instrument to measure that, measure whatever you want to try to find. So for example, I've been using visual analog scale throughout this lecture. So is a visual analog scale one? Is it been validated as a tool to measure pain? And then there should be publications on that. And is it pragmatic to use that kind of scale for this patient population? Visual analog scale, and I just again use an example. So if you're studying a blind population, visual analog scale will be completely worthless, right? Because it is pictures. You need to be able to see. And so you have to take those things into account. Then you develop a plan for data collection and analysis. And I'll tell you, I've got a master's in clinical research, which includes statistics, but I'm not a statistician. I don't have the expertise. I don't do it on a daily basis. I have a working knowledge of statistics. And so you need to get people involved that understand how to design your study from a statistical perspective, which may be different than the design that you're thinking about. Because ultimately it needs to be analyzed. And if you're collecting data that can't be analyzed for some reason, then that's bad. So look, if you can find a biostatistician, that's wonderful if you have access to one. And what they could do is they can also do what's called a priori power analysis. And power analysis are also important, because if you're underpowered, meaning you don't have enough patients in your study, then whatever the results are totally meaningless. So those power analysis it's a simple calculation, actually. It's just an equation. You need to do that before you even start to understand how many patients you need in each group. Then you write the proposal or protocol, attempt to receive funding if necessary. Not all research, in fact, most research doesn't require funding. I would say, 80% of my 200 publications are not funded. And so, most of what you can do, you don't need funding for. Then you implement a research plan, which is like the protocol. It's more practical issues like, okay, where's the lab? Which lab do we send it to? If you're collecting samples, for example. And do you need a pilot study? Maybe there's not enough research to actually, find out where the voids are, and maybe the design that you're looking at is not, it's not crystal clear in your mind. So you do a pilot study, which is a smaller version of the study. And a pilot study also gives you a way of calculating the power that you need to see if there's significance in your outcomes. And then you interpret the research outcomes, and finally you need to communicate those findings. And I'll tell you, communicating the findings are just a lot of people. What happens is they submit an abstract for ACFAS, right? And you put in the ACA, and then they submit the abstract and they present, and it's great. It's good study. It's good early data. But then it's over. It's like, where is that study? And I look for those. I'm one of the judges for ACFAS, the ASC meeting, and I see a lot of good good posters. But then I look for the paper, and I know it's going to take a while for the paper to come out, maybe a year or two, but they just never come to fruition. I always scratch my head like, what happened to that poster? It should have been a paper by now. So the paper is where you want to get to. Don't stop at presenting early data, orally or in an abstract session or in a poster. It goes beyond that. You got to go and publish it, or else you're not sharing it with everyone. So in conclusion, have a plan for a proposed project. Research design is critical. Key to understanding research execution is understanding the literature and reading the literature. The more you read the literature, the better researcher you will be, because you'll pick up on how things are done well and how things are perhaps not done very well.